I don't know why it's... Yeah, I can't hear you. There we go.
There we go. Yeah.
All right, there we go.
What I was trying to say is I now know how to destroy a meeting. You log in twice.
Oh, yeah. It's like yeah. Yeah. So, yeah, I know there was a problem with the timing. I had to change the time quickly before the beginning of the meeting, but sorry about that. I don't know how many people are going to show up today, so because of that quick change in time. So, welcome to the meeting. I know Mainak has some things he wants to share, and I don't know who else is going to be here. I have some things to talk about as well. So, I've got a couple things here that I wanted to share with everyone while we were working on this. Let's see. Is there someone playing video while this is going on, or? Okay. So let me share my screen. Okay, there we go. So today I'm going to talk about a couple things. I wanted to go over a couple things while we're... I know no one's really here, but first of all, congrats to Shruti and Krishna for their acceptance in the Neuromatch Academy. So they've been accepted in the Neuromatch Academy for the summer. Shruti's been accepted as a attendant, and Krishna has been accepted as a TA. So that's he's going to be the TA for the deep learning group, and then Shruti, I guess, is going to be attending the core NMA session. So there's an NMA core session, which is July 2021. Then there's, I don't know, I've not seen this before, this minis thing, which is a half or full day workshop. So I don't know what that is. And then there's the deep learning session, which is deep learning curriculum for 3 weeks. This is what Krishna is going to be a TA for. So congratulations to Krishna and Shruti. I also wanted to point to this. I talked about open source stuff last week. I wanted to point to this database. And this database is for movement data. So this is the movement.openworm.org. And this is, it's something that's managed by OpenWorm. It's the movement validation group. So what they're doing is they're taking videos of different experiments and they're putting them into this platform. They're taking the data, they're putting it into the platform, and they're sort of aggregating it for people to use. So there's a lot of interesting data in here. The samples are hosted on YouTube. You download a video, you can then go ahead and analyze it. They have a lot of summary data here, but they have you can. I think, I don't know if Mayuk may have used it last year for some of his stuff. It may have been, it might be useful to Mainak. It might be useful to other people working on some of the stuff for the worm. Just in terms of machine learning alone, it's a kind of a valuable resource. I know there was a paper published on this recently where they were using the some of this data, and it wasn't an OpenWorm affiliated group, but they used a lot of the data from this database to do their paper. And I don't have the paper on open right now, but I can pull it up for next week and we can talk about it. So that's something else to keep in mind. This is movement.openworm.org, just in case you want to know more about it. Okay, hi Abhishek, how are you? All right, so that's for that. I also wanted to mention that we have our poster that's being submitted to the International C. elegans meeting, and this is the OpenWorm poster. So this is a poster that has all the different sub-projects in OpenWorm, not all of them, but like a sampling of them. We have, for example, the cybernetic project, which we talked about last week. We have the introduction, which is just an introduction to the platform and the project, in the foundation, and the different, just the basic information talking about the Docker file. Then you have this data management, which is an initiative which is OpenWorm, Meta, and ChannelWorm. So they do a lot of data management, formatting data. And then there are the neuronal simulations that are NeuroML is a platform that they use for this. So C302 is the neuronal simulation platform. And NeuroML is used as sort of a basis for a lot of that work. There's the simulation stack in the middle. I don't know if you can see, if I can pull it in a little bit more. So a lot of this stuff is, this is how the current Docker file looks. So you have this Docker image, which you can download to your computer. You can open it up and run it. And these are the current things that it's running. So it's running on, you have to run it on Ubuntu or some Linux platform. You need OpenCL and Python. And then you run the following simulations. You can run a cybernetic simulation. You can run C302. You can run OW Meta, PyNeuroML, and Neuron. And so you get these two are languages that's dependent on, and these three things are what's running. And so you're going to generate, for cybernetic and C302, I think you're generating a bunch of images that end up getting, there have been some problems with getting the Docker container to run without being obtrusive in terms of memory. So, but these two simulations generate a bunch of images that are saved on the local machine, and you can actually go through these simulations just by pressing a button. So that's a nice feature of our Docker container. And again, I'd like to put a DevoWorm component in here, but I don't think we've really found the right component. We've talked about simulating, having a simulation of the embryo. We've talked about having other types of things that are, that we might end up using. But that's going to be, we can continue that conversation later. So then there's Geppetto, which is the platform that enables visualization and simulation of neuroscience models. So this is a bit broader than the neuroscience models that we use in C302. This is actually something that we can integrate with things like Drosophila data and other types of things. So Geppetto is actually run by, it's kind of semi-independent of OpenWorm as well. And the WormSim website is where you want to go to find out more about that. And then finally, our contribution is here. This is the DevoWorm contribution. This is where I just kind of talk about the different, I think they've put XXX in here. They're going to be renumbering the references, but so, in this one I put a little triangle of stuff. So this is the connection to OpenWorm, this is the DevoWorm group, and this is the open data component. Then I put this component in, which is the simulations that we have from DevoLearn. We have characterization of our 5-tuple, our 5-dimensional data space, the 3D space plus time and context, this theta, which can be a number of things, and I've talked about that in previous meetings. And then the DevoLearn logo. So we're, talking about DevoLearn, we're talking about some of the work that we've done with comparative biology and then complex networks, which is, there's an image in here. It's hard to see at this magnification, but there's a embryo network in there. So, and then I put a couple of references in here. So hopefully that's a good representation of DevoWorm to the this is to the International C. elegans meeting, and this is going to take place online. I think this month, We have like maybe 2 more weeks until it happens. So hopefully it's well received. I did a DevoWorm poster at that conference, I think in 2015, and actually I met up with Steve Larson there. It was held in Los Angeles, and he was in San Diego at the time, so he just drove up, and then we met there, and that's the first time I had met him in person. And then I met him again in the same year at another conference. So that was I haven't seen him since then. So in the flesh. So that's I don't know. We still won't be able to meet in person. So that's the thing there. Okay. So that's all I have for that. Now I wanted to invite Mainak to give an update on his stuff.
Yeah, I'll just share my screen and
Okay.
I'll do it right now. Okay, so can you see my screen?
Yes.
Hello. Okay, so first I'll go over the weekly update and then I'll go over the coding period timeline that we have. Okay.
All right.
So okay, I'll get into it now. Okay. So this week I basically spent a significant amount of time trying to find some new data to work with basically, and I actually browsed through a lot of papers, but I came across this specific paper, and this paper actually led me to a website which contained a lot of data. So this is the paper that I'm talking about. It says 3D convolutional neural network based segmentation to acquire criteria of the nucleus during mouse embryogenesis. So it's pretty long, but I'll skip to the part that I used. Yeah, so in the data availability section of this paper, they have linked to the Cell Tracking Challenge website, which is essentially an open, it's basically an open competition on the internet, and anybody could go ahead. And this is the data set I found. So this data set basically contains C. elegans embryo fluorescence microscopy data with segmentation maps. So what I did is that I basically downloaded the dataset and I basically tried to pre-process it for future use. So I'll go ahead and get into the details of the data now. So the data basically contains feature and label pairs like this, and this image basically is the fluorescence microscopy data, and to the right we can see the segmentation map, and it's actually semantically segmented, which is a good thing, and the format is in TIFF format, and the total size is 3.1 GB, and I actually have to convert this TIFF data into NumPy arrays in order to plot these photos. And if I zoom into the image, and okay, I'll just zoom into this image because I wanted to add something to it. Okay, so in this image, like these little images, they basically show the slices of 3D data in form of these 2D slices. So as you can see, like in the extreme points of the axis, we can see that most of the data is not really usable. Like we have got a lot of blank frames to the top and to the bottom. So I was thinking of getting rid of these data points entirely, but I'll see what I can do with that. But for now, I'll just keep it. So the model, I'm actually expecting expecting the neural network to actually learn what's going on, and I think it should also learn to classify these empty points. So okay, so as I had said that the data was in a TIFF format, so I basically had to convert them into a JPEG format for ease of use, and I stored them in this Drive website, this Drive link, and there's also the Colab notebook, and so if anybody wants to check out the code and just add stuff to it, they could go ahead. And okay, so the work is also stored in the GitHub repository of the GSoC 21, and the Colab notebook is also there as I showed. So what I plan with this data is to basically train a neural network to take input of 3D data in form of these slices, and then have the model basically map it to these 3D segmentation maps. I know it's a bit far-fetched now, but I'll see what I can do. So coding period actually okay. So I guess that's it for this week's update.
Okay.
But now I'll be talking about the coding period timeline which is proposed. So I think I'll have to zoom in now because it's hardly visible. Okay, so is the text visible?
Yeah.
Okay, so the first week which starts today. So for the first week, I basically have plans to refactor all the models according to the inference engine class, and then inspect the data sets, then take some feedback. If someone has to add anything, I'll just go ahead and get in touch. And also I'll have to pre-process the expanded data sets, which I talked about in the proposal. So the second week, I have kept it for the embryo segmentation model, and I have actually described the details in these points. So the third week, it's for the lineage population model. I'll be upgrading the lineage population model, and I'll be using the expanded data sets that I had created. Not created, actually compiled. So, okay, so the fourth week, it would be spent mostly on creating a report of the training metrics, and this report will essentially show how far the improvement has come, like how good the improvement is compared to the older model. So I'll also be adding a new function which would enable the users to run inference on NumPy arrays. And I found this idea on the Slack channel because somebody was talking about this in the Slack channel, and I basically took that idea from there. So I also have plans to add an argument to add the plotly plots instead of the matplotlib plots which are static. So that would be a good addition, I guess. And the fifth week, which would be the final week before phase 1 evaluation, that would be spent on creating a summary and a report and uploading everything to my blog. Plus I've kept a few days at hand in case I run late and in case I have to work a bit more. So that's for phase 1. And for phase 2, we have 4 weeks in total. And in those 4 weeks, in the first week, which is week 7, I'll explore the EPIC 2 dataset. But I think after I found the cell tracking challenge data, I think that could also be replaced by this. Like I could also work on I could also replace the EPIC 2 data with that dataset. So I'll see what is to be done there. And in the following weeks, I'll explore some dimensionality reduction techniques like UMAP and some other projection techniques, and I'll try to extract some patterns or some sort of a meta info that if I can get it, and then I'll upload the training notebooks, which is very important because like for starters, it's very difficult to get any idea of how these models are trained. So I'll upload every training notebook that I create, and I also have planned to create a Docker file of the DevoLearn package. And then for the interactive demos that I talked about, like for the interactive demos, I plan to set up a DevoLearn blog. Like it would be a very simple site with some minimal interactive demos, like which would have sliders, and you'll be able to change the input, and you'll have the interactive. You'll also be able to see the interactive outputs to the side. So that could be done, and yeah, and the only documentation that DevoLearn has right now, it's basically the README file. So I think it requires a read the docs. I think it requires a formal documentation on the internet, and I think a read the docs or a Gitbook could be used for that. That could also be done. And yeah, the final week I have left a couple of days in case I'm late, and I'll also update my blog and the reports. And then yeah, and I guess that's the end for the timeline. So I'll try to stick to it as much as I can, and I'm pretty sure I'll be able to get everything done.
Right.
So like if there's any feedback, anybody could go ahead, and that's it for my presentation this week. Thanks.
Okay, thank you. Hello, Shruti and Jesse. How are you? That's very good. I want to talk about a couple of the slides. So if you could go to number 2.
Yeah,
Okay, so
sure.
the hunting for data sets. So you said that there was this cell tracking challenge. So
Yeah,
what are the data sets there? Can you show? Can you go
yeah,
to
sure,
the link?
sure. Yeah, so the cell tracking challenge, like the homepage contains a lot of data sets. Like there is 2D plus time data sets and 3D plus time data sets. And this is the homepage. This is actually a competition online. I actually came across it very recently. So what I did is that I went to the 3D plus time data sets because that's the only page where there's any 3 elegans data. Because okay, so these are the data sets that's available. Like I'm not really familiar with all these other data sets, but what I found interesting is this one. So
Yeah.
this is the dataset I downloaded, and it actually contains 35 planes for each time point as data. So I think that's enough for training a model, and I'll see what I can do. And it's actually pretty big. It's 3.1 GB,
Yeah.
and training data. And if it's 3.1 GB of training data, and it actually comes down to almost 10,000 data points. So I think that's a lot.
Yeah.
Plus, plus like if anybody else actually wants to find some other data sets, they could find it here, and the data sets are actually like they're good, they're not bad. So
Yeah, they're
yeah, that's
different
good.
systems.
Good thing that I came across.
Yeah.
Yeah,
So
and
the-
plus like if anyone wants to know the specs of the data, like how they captured the data, that's also given here, like the voxel size, the objective lens. I'm not familiar with all this, but I don't think that's required, but it's there, which is a good thing.
Well, it tells us the acquisition. But yeah, going back to
Yeah.
so this is the same data set as the Epic data set, more or less. It's the same group.
No,
Well, it's the same group
Yeah, sort
that
of.
they're doing. Yeah. I mean, so
but the
they're
epic data set. Okay, so can you tell the basic difference between the epic data set and this?
yeah, yeah.
Yeah. So the epic data set, it really doesn't have any segmentation maps to go with it.
Okay.
So what we have here is that for each time point we have these slices. So this essentially is for one single time point, and these 35 slices they basically represent the data for one single time point, and this represents the segmentation map for that time point. So, but the epic data set, it contains other data. It really doesn't have any sort of segmentation data to go with it. So this is the advantage of this data set compared to that. So I guess it will be. I don't know. I'll explore this further and I'll keep you guys updated with that.
Yeah, it would be great. Yeah, that's great. And so it's from the same group, so they use sort of the same methods, so it's like a good comparison, because sometimes you get variation between groups, and but yeah, it's so that's good. And then we have the semantic map, which is of course valuable, and then the data here. So we have the segmentation maps. Yeah, those are great. You talked about this in the Slack channel. So okay.
Yeah. And I also have to convert the data into another format for ease of use, because TIFF format, it's not really like, it's converting them to NumPy arrays, it's actually a bit slow. So I actually converted them pre-hand, so the training is faster, and I'll see what I can do with that.
Okay, that's great. Then you have what else? It's the second to the last slide. Oh, your schedule, right?
Yeah, this is the timeline.
Yeah. So I guess that looks good. It's going to be the thing you're going to have to watch out for is you're going to have a lot of things in 1 week. So, you want to make sure that you kind of, you want to keep track of time, but you also want to get stuck on anything. If you get stuck on something in week 2, you want to either say, okay, I'm going to finish this up in 3 and then, modify it accordingly, or, sometimes you just have to say forget it and then move on to something else and then come back to that later. So that's what I'm getting at with the schedule. But I think at the end you have this week 9 and you say set up a DevoLearn blog, which would serve as a hub for users. So this is something that so it would be like a blog out of GitHub.io, or would this be something that is separate? Because we have we have a GitHub IO presence already. I don't know. We might want to keep it on that in that space. Like we have the Devo Zoo and some of the other stuff there. That would be good.
So maybe we can have a separate branch for this. That could also be done.
Okay. And yeah, I think the
We
idea
have a DevoLearn section and
I think that the idea of having these little demos of sliders is great. We haven't gotten to that stage really yet, which is because basically but I think that would be great to have, especially as an interactive tool. We try to do that sometimes in the notebooks, but sometimes those are not they're not always accessible to people. So I think doing a web interface would be good.
Yeah. Plus, I'm pretty sure I'll need a bit of help from the other developers in this community because I'm not very experienced in web, but I'll try my best. That's all.
Okay. Well, I think Ujwal did some web development in his project last year, so he'll be able to help out.
Yeah, sure. I'll get in touch with him then.
Yeah. Okay. Any questions from anyone else about the Mainak's presentation? I think it's probably pretty good. Let's see. One is, okay, Mainak, can you put your email address in the chat? Dick wants to have it, wants to go over some of this maybe. Yeah, so that would be great. Let's see. So now I'm gonna. Is anyone does Jesse or Shruti or Dick want to present on anything today, or do we have? I'm gonna move on to other things if not.
No real updates from me. It's good to hear everything.
Hey Bradley.
Hello.
Hi Jesse. Hi Dick and hi Janet.
Yeah, again, congratulations to Shruti for getting into Neuromatch.
Yeah.
Yeah.
Thank you, Jesse too, and Krishna also organized the TA.
Okay.
Congratulations.
Thank you.
Yeah.
Okay. Bradley?
Yeah.
Yeah, I could give a very brief presentation on the problem of segmenting images of Archaea and comparing them to shape droplets.
Okay. That'd be good.
Okay. Let me pull up the file. Let's see. Let's see, we gotta find the file.
Okay.
Second. This is an impromptu. Okay, found the file. Okay, now share screen. Okay, share. Okay.
Oh, there we go.
Good. Okay. When you see this paper, Emergence of Polygonal Shapes.
Yes.
Okay, good. Okay. This is a 3-column presentation. These, first of all, let me explain. If you take oil in water and shake it up, ordinarily you get spherical drops. Okay? But if you have a little bit of surfactant present and you cool it slowly, you get these polygonal shapes. That's in the first column here. They're called shape droplets, and you can see they can be triangles, they can be triangles with concave edges, they can sometimes form these weird things on the side, they can be squares, rectangles, pentagons, hexagons, and they've been observed up to 9-sided. Okay, and these are droplets of oil. Now, there's a whole class of organisms called Archaea, which were thought to be the same as bacteria, but they were discovered genomically they're very different, and chemically they're quite different. And they also sometimes form triangles, squares, rectangles, pentagons. Okay? And the third column are diatoms, which are more advanced organisms, but they also sometimes form triangles, squares, 1, 2, 3, 4, pentagons in higher order. Here's a 12. 1, 2, 3, 4, 5. This is a 7 pentagon. Okay? Okay, now here's the problem. We like to hypothesize that the shaped droplets in the origin of life are what the Archaea grew out of. It's called the Archaea first hypothesis, but here applied to the shapes. Okay, so the question is, how similar are these shapes? For example, if you look at the Archaea carefully, you can see that even though this is triangular, instead of a 60-degree corner, they seem to have 90-degree corners. So there might be some differences in that regard. There might be some structure in the corners that makes them 90 degrees. Okay? So here's the problem. I've collected a large number of pictures of these things. And can we quantitate the shapes and compare them and see if how different are the shaped droplets in general from the Archaea, and how similar are they? Okay, so it's a large segmentation and geometry problem, and you can see that the quality of the images is very different. So we need a segmentation that's fairly robust to image quality. Okay, so I'm looking for a partner who is experienced with segmentation of cells to go over the images and see what we can extract in terms of geometric parameters.
Yeah, that would be great.
Okay, that's it.
Yeah. Yeah, so it's, that does remind me of the stuff that we're trying to do with the talk on the TopoNets, where we have these geometric shapes that, we're considering like the sort of the form and the edges instead of the nodes as like informative. And so, to fit that, it's almost like, you could use an approach to like finite element analysis or something similar, where you have all these different, where you're trying to fit a mesh or something to it and to see what the differences in shape are. But yeah, I mean, that's something though we can talk about later.
Yeah, I mean, we have to detect things like number of corners, what's the angle at the corners, how straight are the edges, questions like that. Okay?
Yeah, that would be great.
And there's how uniform are the edges in length? Okay,
Right.
so there's a whole raster of geometric questions to be asked about these things, and then to compare these parameters between the shaped droplets and the Archaea to see if there are any significant, where are there significant differences, and are they significant in terms of origin of life hypothesis or not?
Right.
Okay.
Yeah.
Okay, so that's the problem, and looking for a volunteer.
Yeah, if people are interested, that would be great. It would be something I think we could pick up on, a theme that would be really productive. So yeah, that's great. Let's see, we have some things in the chat. Okay, yeah, so that's linked to Mainak's slides, this week's blog post, which is something that Mainak has to do weekly, And then Dick, can you share this paper? We'd love to read it. So yeah, the paper on that you were just showing on the Archaea. I don't know if it's in press or something. You can't share it.
No, I can't. It's
okay,
published a few years ago.
Oh, okay. So yeah, so all right, then let me get on with the. Here, share your screen. Okay, we can get on with the submissions and things. This is our submissions list, as you've seen before. We have, I think we've cleared a fair amount of this, but we still have other things we need to revisit. So number 8, this OpenWorm poster, I just discussed this. This is to be, I think, presented in about 2 weeks. So I think we're pretty much done with the edits on that. I think that was a good summary of DevoWorm as it stands now. And we just didn't, we didn't have a lot of space to go into things in depth. So that's just, to give people a taste. And I wanted to put a lot of images on there so that people can, in posters, you need to use a lot of images. This stuff for Networks 2021, that's still kind of in progress. That's going to be a push for that later on this month. And then I'm sorry about this non-neuronal cognition paper. This is something that's been really kind of long suffering, and I'm trying to finish it up, but it's been, we're trying to generate a workable paper. And so I talked to, I think, Dick and some other people about it this weekend, and I just sent Dick a version of it, and it's somewhat rough, but I wanted to get some feedback on it. So that's going to be worked on as well this month.
Could you potentially send me a version of the paper too?
Yeah, why don't I do that? I'll send you a link to it after the meeting. Yeah, I mean it's pretty rough, but yeah, if you could provide feedback, that would be great. We could follow up. I mean, it really needs a lot of fleshing out. There's a lot of stuff in there, a lot of ideas. Some of them maybe aren't great for the scope of the paper. Some are maybe very important and need to be expanded upon. So we need to figure out what those are. And so that's that. And then this, let's see. What else is coming up? I think a couple of things. Yeah, there are a number of things that have been submitted that we're waiting on some other decisions. We have these here, the test of Williamson symbiosis and the molecular level simulations of diatoms. Those are still on the list if people are interested. And then this, actually 28 we have on the list, which Dick just talked about with a quantitative comparison of Archaea and shaped droplets. So just to keep on top of that, that's good. What else do we want to talk about? Why don't we just get into papers now, since it gives us a little bit more time to talk about this. But so we've got a couple, several nice papers here. I'm always trying to catch up on the newer papers, but this is a good opportunity. So the first one I'm going to talk about today is the synthetic living machines, a new window on life. And this is I think it's a new paper for Michael Levin and a collaborator. Yeah, this is a newer paper. So, he does this stuff with xenobots, which are these robots that are cells, essentially. And they're using synthetic biology to sort of program those cells. And so that's a big area that has gotten a lot of press. So I think this builds off of that. They're making the argument here, synthetic living machines, a new window on life. So the summary is increased control of biological growth and form as an essential gateway to transformative medical advances. Repairing of birth defects, restoring lost or damaged organs, normalizing tumors, all depend on understanding how cells cooperate to make specific functional large-scale structures. And we've talked about that with respect to development and, regeneration. Despite advances in molecular genetics, significant gaps remain in our understanding of the mesoscale rules of morphogenesis. So what mesoscale means is that it's not like the interactions between molecules and it's not the type of like tissue formation, but it's this or morphogenesis over the course of all of development. But these sort of mesoscale rules, which are in between those, and so we don't really understand. We understand a little bit of how it works, but not entirely. An engineering approach to this problem is the creation of novel synthetic living forms, greatly extending available model systems beyond evolved plant and animal lineages. So this is, this is just creating sort of new model systems instead of relying on the typical model systems that we use. Here we review recent advances in the emerging field of synthetic morphogenesis, the bioengineering of novel multicellular living bodies, emphasizing emergent self-organization, tissue-level guided self-assembly, and active functionality. This work is the essential next generation of synthetic biology. Aside from useful living machines for specific functions, the rational design and analysis of new coherent anatomies will greatly increase our understanding of foundational questions. So they get into this. So here's figure 1, and they kind of talk about, again, this is one of these broad diagrams that kind of explain what they're getting at here. So they're sort of founding this new area here, synthetic functional morphologies. So, this requires a number of different disciplines. It requires biology, it requires us to understand biophysics, developmental biology, and even evolutionary biology. It requires computer science, in this case machine learning and swarm robotics. And swarm robotics is, where you take a bunch of robots and you try to get them to behave like social insects, basically. They'll, communicate with one another. They have these means of like forming swarms at a desired time around maybe, you provide them with computational resources, you provide them with like cues in the environment, and they behave like a collective. And so that's important. Machine learning is also important. There are probably other areas that aren't mentioned here. Also synthetic morphology, cybernetics, and cognitive science. And of course we know Mike Levin's view of cognitive science and cybernetics and how that relates to biology. We talked about that last week. As for tools, we want to, this is, I think this is related to an organoid model, which we've talked about. So you can use things like optogenetics, which is where a fluorescent protein is put into a cell, and it fluoresces according to some stimulated frequency of light. And whenever you stimulate the cell, it shows this light, and it shows whether there's a gene being expressed or not, and the activity of molecules in the cell. You also need to know organoid culture, so you need to be able to culture organoids, nanomaterials, and computational model and in silico evolution. So this is like things like digital evolution or simulating cells in a computational environment. So that's a lot of tools to master and a lot of disciplines to bring together. But the impacts are very useful. You can use these kind of models for drug discovery and disease modeling, decoding design principles of human development, the evolution of body plans, which we've talked about in the past, regenerative medicine of birth defects, injury, and cancer, new AI platforms and algorithms, swarm robotics, and useful biobot functions. And so these are all things that you could use. These are very speculative, especially the swarm robotics aspect, where you're basically using it to get cells to collaborate with one another and communicate. So this is a pretty forward-looking paper. They're talking about things like engineering synthetic multicellularity, and there's a paper here from Ricard Soleil that talks about that. He's done a lot of work with complexity and bioengineering, and so here's an example here of synthetic embryo-like entities. So we have synthetic embryos, or these embryo-like entities. They're called them blastoids, and they're co-stained with F-actin here. You can see in nano green, which is in here. So you can see in this, I think in this figure here, nanog and F-actin are expressed in different parts of this synthetic embryo. Let me bring back this paper. There you go. So you can see it kind of looks like the blastocyst of a normal embryo, but they're blastoids in that they're created from these cells that are synthetic cells, and they come together and they're expressing the types of molecular markers that you might see in a regular embryo. Then they show this here where it's a schematic to show strategy for generation of mouse embryonic-like structures, combining the mouse, which is the red component here, and the TSCs, which are the blue, which are these specific type of stem cell here. So these are mouse ESCs, these are TSCs, and these are the, so they're culturing these different types of cells in different contexts, and then they're combining them into a single organism, or a single entity, I should say. And then they have these gastruloids at 120 hours at the bottom after aggregation, showing features of a mouse embryo on embryonic day 1. And then you start to see spatially confined signaling cues and markers, which means that it's behaving sort of like an embryo that's differentiating into different parts. So these gastruloids are just these mixtures of two types of cell. They're coming together and then they start behaving like a real embryo. So this is like a nice example of what they're actually trying to do here. They're trying to create an embryo, and we're going to actually talk about another paper where they do the same type of strategy, where the embryo isn't necessarily an embryo as a whole, it's actually just the behavior of cells in the embryo. So this becomes important when you're trying to create synthetic embryos that have, that they're just trying to assemble from scratch. So they're able to do this. They talk a lot about organoids and synthetic tissues, being able to create tissues that mimic human organ-like structures. So it's just basically a review of organoids. And again, you have this examples of organoids in multicellular tissues. So this C is a schematic for genetically guided engineering using stem cells, engineered with inducible genetic circuits to pre-program cell fates associated with different germ layers. So this one here is where they've been able to take like different germ layers in the embryo, combine them into this aggregate. And so let's see what else they have in here. So this is where they talk about biobots and xenobots, and so this is one of their things that they're doing. It's getting a lot of attention, especially in the alewife community, where they're able to, this is an artificial ray that they've created using a 3D elastomer body, and they're able to generate a skeleton and then muscle. So they're able to build these layers of tissue on top of this artificial scaffold, and so they're able to assemble this artificial ray, and the ray can actually do simple behaviors by using optical stimulation. So that's this technique that I mentioned before, where they can just take like a light signal at a certain frequency and stimulate the cells in a certain way. They can show what's being expressed in the cell, but they can also make the cell do things. So they can make the cells excited, or sometimes they can drive movement of cells or coordinated movement of cells. In this case, I think they're actually able to move the ray in a certain direction by stimulating the cells with a certain light frequency, and then they're able to induce muscle contractions and move it forward, or to make it undulate its rays or its fins here. And so they're able to get it to maneuver. So in a couple other examples here, this F are the xenobots modeled in a virtual environment. So they're able to actually model this in silico to show how these things can be assembled and the structure of the system. So that's a lot of work. I think this artificial ray is not really something that they've created. It's in the planning stages, but that's the idea of what they're trying to do here. And so just to be clear on that. this is an example of extracting cells from xenopus embryos, allowing these cells to reassociate, and then microsculpting to remove some of the cells. And that's how they basically try to form some of these xenobots. They take cells out of context, they put them in a new context, and then they shape them. So this is so it requires a lot of genetic engineering. Here they're trying to engineer gene circuits, and they're trying to control what they call pluripotency, which is this ability of the cell to shift its fate or to make fate decisions based on some stimulus that they're giving it. So cells start out, actually the embryo starts out totipotent, and then it becomes pluripotent. And then you have pluripotency where you can switch between different cell fates. So if you're a pluripotent stem cell, you have a restricted set of fates, but you can change those fates using some sort of signal. So like you can take transcription factors, or you can take chemical inducers, and you can switch the state of the cell from a stem cell to a differentiated cell. Sometimes you can go from a differentiated cell to something that looks like a stem cell. But this is how they're able to deal with a lot, how they're able to sort of manipulate the embryo or the gastruloid. So there's a lot of stuff that's involved in this. I'm going to put this paper aside, and let me give you the link to the Drive folder here so we can... Okay, Shruti had to leave early, thank you. And Jesse had to leave as well. But we're going to go through one more paper here. This is the one here that I was telling you about, where in this case they're actually arguing, in a sense, that you don't need to have a whole embryo to get embryogenesis. You can actually use stem cells, and you can use things that aren't embryos to create an embryo or to control embryogenesis. So this is construction of a mammalian embryo model from stem cells organized by a Morphogen signaling center. So the abstract here is that generating properly differentiated embryonic structures in vitro from pluripotent cells remains a challenge. Here we show that instruction of aggregates of mouse embryonic cells with an experimentally engineered Morphogen signaling center that functions as an organizer results in the development of embryo-like entities, or embryoids. So they're using this embryoid method again, where they're trying to create, they're taking cells, or putting it in a culture, and they're trying to create something that's like an embryo that has the capacity to sort of form and differentiate like an embryo. But they're actually cells that are put into this context separately from an embryo. In situ hybridization, immunolabeling, and cell tracking, transcriptomic analysis, so these are all analysis methods, show that these embryoids form 3 germ layers through a gastrulation process, and that they exhibit a wide range of developmental structures, highly similar to neurula stage mouse embryos. So these are basically cell cultures with a bunch of stem cells or pluripotent cells in them, and then they can sculpt those cultures to behave sort of like an embryo. They're able to coordinate the differentiation patterns there. Okay, so embryoids are organized around an axial choromesoderm with a dorsal neural plate that displays histological properties similar to the murine embryo neuroepithelium. And that folds into a neural tube patterned anterior-posteriorly from the posterior midbrain to the tip of the tail. So there's this patterning along the anterior-posterior axis. In this case, they get this sort of neural tube that forms, and it's forming from sort of the middle part of the organism, maybe the midbrain, which is sort of at the anterior end, and then to the tip of the tail, which is the posterior end. Lateral to the choromesoderm, embryoids display semitic and intermediate mesoderm with beating cardiac tissue anteriorly and formation of a vasculature network. Ventrally, embryoids differentiate a primitive gut tube, which is patterned both anterior-posteriorly, which is front to back, and dorsal-ventrally, which is top to bottom. So you have these different you're getting these different anatomical axes, and so the front to the back, the top to the bottom, and they're actually getting these things that look like a primitive gut tube and a neural tube, but they're not. I mean, I don't know how. If they're actually going on to form, they're not going to necessarily go on to form a fully formed organism, an adult organism. So, altogether, embryoids provide an in vitro model of mammalian embryos that displays an extensive development of germ layer derivatives, and that promises to be a powerful tool for in vitro studies and disease modeling. So we have a nice model here, and again, this is where you don't necessarily have to have a full embryo to have embryogenesis. You can take a bunch of stem-like cells and put them into these organoid, form these organoid structures that actually form, or I guess they call them embryoid. I think the name varies by what they're trying to do. It's always at the ending oid. So they have embryoids and organoids, but they basically grow them in culture and they can manipulate them there. One limitation that may explain why only partial development is obtained in vitro may be related to the method used to induce developmental programs in embryonic stem cells. In vertebrates, the signals breaking the initial spherical symmetry of the egg and instructing cells about their fate and behavior are present in the egg or are generated by the cells of the embryo itself or by its extraembryonic tissue. So you have all these things that happen in the mouse embryo. You have a primitive streak, extraembryonic ectoderm, and visceral endoderm in the mouse embryo. So these are things that are signals that break that initial spherical symmetry. And you may or may not see them in vitro. Therefore, the developmental programs that control patterning and morphogenesis are not induced by soluble signals present in the liquid medium surrounding the embryo. So that means that you have this, there's obviously something going on in the embryo that you don't necessarily have in vitro, although you can see these things form in the embryoids that resemble what's going on in the embryo. So they're able to do, they have these protocols that they can use to sort of induce some of these chemical signals, but So let's see if they have any figures in this paper. So this is a pretty hardcore picture here. This is where they just talk about their protocol. They show some of the things that they're doing. They do this hanging drop method. Then they merge the culture at day 3. I can't remember what a hanging drop method. I've done this before, but I can't remember exactly what it involves. But that's, yeah. So they form these small aggregates and these large aggregates. And so they're able to sort of figure out signaling centers.
Bradley?
Yeah.
You hear me?
Yeah.
The hanging droplet is very simple. You take a cover slip and you put it, you use it with a microscope slide that has a spherical well that's been carved out of it. So there's some space.
Ah, right, yeah.
You drop on the cover slip, a drop of liquid on the cover slip, and you turn it upside down so it hangs.
Right, okay.
Okay?
Yeah. So these are the formation of embryoids by instruction of VSC aggregates, or the Morphogen signaling center. So here they have these small aggregates and large aggregates. They induce a signaling center somewhere in the organoid, and then they watch they monitor it over a number of days to see what happens. And it turns out that they get these different things that differentiate with a signaling center involved. So this is position over time of mesoderm cells during the gastrulation of embryoids. Maybe this is a better figure for kind of what we want to talk about here. So this shows, I guess these green cells are these are broad GFP expressing cells. So this is their marker for the mesoderm cells. And they're moving towards the anterior end of the gastruloid. and there's sort of this wave over time that's driven forward, but also driven sort of forward by Winton nodal expression in the back end here that's driving them sort of forward, and then they come back. So there are all these different types of things going on with this. So let's see. C is an interpretive drawing mesoderm cells induced by Wnt3 nodal, which are these blue arrows here. In the signaling center were observed progressively more anteriorly during gastrulation. So this red arrow is the direction of travel, and you can see these green cells that are moving forward. Winton nodal expression is moving forward as well. In F, we have interpretive drawing, red arrow, change in position of BRA-GFP. So this is where they're changing the position of BRA-GFP. Winton nodal are secreted by unlabeled cells of the signaling center, induced the expression of BRA-GFP in a stripe of adjacent cells deriving from the naive aggregate. So they're changing the position of where the signaling center is, and they're able to get differences in these, how these things are expressed spatially. And so this is just the kind of stuff they can do with this kind of model to sort of get a handle on some of the signaling pathways and that, and how they behave in these aggregates. Okay, so I'm gonna stop there, and I think that's all for today in terms of papers. So again, if you have anything you want to present, just let me know. Also, I have some papers from Dick that he sent me on various topics. We'll probably go through some of those in the next few weeks. And if in Mainak, keep up the good work and keep presenting on. I mean, we'll have an update every week, I think, on your work about the same length that you gave this week. So we'll just kind of keep reviewing your work, and hopefully you stay on top of everything. And if you have it, again, let's keep in touch on Slack all week. Make sure that we if you have any problems, we can address them quickly.
Hey, Bradley, do you have a minute after we're done to discuss the stuff that Janus Pappas?
Yeah. Okay, so thanks for meeting. Have a good week.